In this module, we will discuss the role of protein tyrosine kinases in the mechanism of cell signaling. And we will discuss that how protein tyrosine phosphorylation as a mechanism of signal transduction occurs and what are the receptor dimerization. Now, protein tyrosine kinases, they are the enzymes that phosphorylate specific tyrosine residues on protein substrates. Remember, we have been discussing constantly the role of enzymes in the cell signaling. And the protein tyrosine kinases, they, as a matter of fact, they control the signals in the growth, division, differentiation, survival, and attenuated to the extracellular matrix. They are attachment of these enzymes to the extracellular matrix, extracellular cells, and as a matter of fact, they also control the signaling of the migration of the cells that occur during the growth. Now, they are divided into two large groups. One is the receptor protein tyrosine kinase, which is abbreviated as RTKS, and non-receptor or cytoplasmic protein tyrosine kinases. These are the non-receptor, they occur in the cytoplasm of the cell during the signaling. Now, how receptor dimerization occurs? It is the presence of a growth factor on the outside of the cell, which gets translated into the biochemical changes. Remember, the cell signaling is always connected with the biochemical changes. The biochemistry of the cell, it changes, and that can be a ligand binding that results in the dimerization of the extracellular, जो cell के बाहर हैं, that ligand binding, and the domains of the pair of acceptors, that is, जो पेयर हैं, जो जोड़े हैं, जो के ग्रुप्स हैं, उनको एक्सेप्ट करते हैं इस एंजाइम से. Now, the mechanism for receptor dimerization will be two type. One is, as already said, कि ligand mediated dimerization. Ligand का मतलब यह है कि chemically they are mediated, and the second one is the receptor mediated dimerization. इसका मतलब ये हुआ कि जो ligand mediated हैं वो तो chemically हो गए, और receptor mediated dimerization, that will be the development of some receptor on the cell which control this. Now, the ligand of RTKs, it contains two receptor binding sites. इसकी दो sites हैं, दो जगह हैं जहाँ पे ये bind कर सकता है. And this is made it possible for single growth और differentiation factor, कि इसकी growth जो है उसमें या differentiation, कि किस तरह ये differentiation होती है, उसमें ये bind करते हैं to the two acceptor at the same time. And thereby, when they are attached to the acceptor at the same time, what they do? They cause the ligand binding or ligand mediated receptor dimerization. Dimerization का मतलब ये एक monomerization है जिसमें monomer है, और di में ये है कि they are two. However, not all growth factor they appear to conform to this model. सारे जो growth factors हैं वो इस model को confirm नहीं करते, इस model को follow नहीं करते. Some growth factor such as EGF, TGF, they contain only a single acceptor binding site. उनकी एक ही acceptor binding site होती है जहाँ के वो bind करते हैं and control the cell signaling. a second mechanism where ligand binding induces a conformational changes. Remember, whenever there is a ligand binding, there will be a conformation changes. The conformation changes means that is the change in the protein molecule, in the protein extracellular circle or a domain of receptor that lead to the formation or exposure of receptor dimerization. कि जो एक्सेप्टर है, जो रिसेप्टर है, उसकी डाइमराइजेशन जो है वो चेंज हो जाती है. रिसेप्टर डाइमराइजेशन इसका क्या होता है? इट रिजल्ट्स in juxtaposition of two protein tyrosine kinases domain on the cytoplasmic side of the plasma membrane. Means that the tyrosine kinase proteins, they juxtaposition mean एक दूसरे के साथ जुड़ जाती हैं, एक दूसरे के साथ बाइंड हो जाती हैं, so that in the cytoplasmic side of the plasma membrane, and then they control the signaling of the cell mechanism.